Good evening, guys. Today we'll be starting with the last lecture of the RNA biology classes. So, today we'll be discussing about different previous year questions related to translation and post-transcriptional modification in eukaryotes that were asked in various examinations. And if there are any doubts, you can let me know. Okay. So, starting with the first question. The following statements were made about X chromosome inactivation in humans. So there are statements given. So it is maternally derived X chromosome have a greater chance of becoming deactivated in any given cell. Both X chromosomes are activated determining the process of oogenesis. The exist gene encodes for a single non-coding transcript which binds with the X chromosome and helps in its inactivation. The excess gene expression is required to initiate inactivation of X chromosome and also to maintain inactivation from one cell generation to the next. T6 transcription affects the abundance of excess RNA in cis. Okay, so which one of the following options represent the combination of all correct statements? So as you know that X chromosome inactivation. So all the females invertebrates have all the vertebrate females have two X chromosomes, and out of these one is inactivated. So this is random process. It's not the case that maternal chromosome or paternal chromosome will be inactivated. So it's a random process. So the first option states that maternally derived X chromosome has a greater chance of becoming inactivated in any given cell. So this option holds false. Okay, so the first option is false. Then both X chromosomes are inactivated, denying the process of oogenesis. So, in the second option, demine means when, like in the process of oogenesis, before the process of oogenesis, both the X chromosomes are activated because there are equal chances of either of the X chromosome to be distributed in the daughter cells. So that's why the both chromosomes are activated, denying the process of oogenesis. So this option is correct. Then we have the third option. The excess gene encodes a single long non-coding transcript which binds with the X chromosome and helps in its inactivation. So yes, as we remember, so this was the X chromosome. Here it produces a long non-coding RNA that is excess, so it goes and binds to the X chromosome that is being inactivated. Okay, so option C is also correct. Then we have option D. So excess gene expression is required to initiate inactivation of X chromosome and also to maintain inactivation from one cell generation to the next. No, this is not correct because the inactivation pattern does not transfer from one cell to the other. Okay, so it remains for that particular cell only. The T6 transcription affects the abundance of excess RNA in cis. Yes, this option is also correct because the T6 RNA, it inhibits the expression of the excess RNA, and this T6 RNA basically inhibits breakdowns the excess RNA. Okay, so this is the inhibitor of excess RNA. So options B, C, and E are correct. So option 2 is the correct answer here. Okay. Now moving on to the next question. So with respect to X chromosome inactivation in females, which of the following statements is incorrect? So X inactivation first occurs in the pre-implantation female mammalian embryo. Okay. So it yes. So what happens before the embryo is implanted? So um, Then the second option states one of the two X chromosomes in each diploid cell of a normal woman is randomly selected to undergo X inactivation and becomes highly contagious. The process involves epigenetic silencing of each gene on one of the two X chromosomes, either the maternal X chromosome or the paternal X chromosome. Once the decision is made to inactivate a paternal X or maternal X in the cell, all descendant cells will continue with the pattern of X inactivation. Okay, so starting with the first point, so X inactivation first occurs in the pre-implantation female mammalian embryo. So yes, this option is this option is correct because when the like embryo is formed. So this is the sperm, and here we have the egg. So when the fusion occurs and a zygote is formed, so before pre-implantation of the embryo, it has been decided that which X chromosome will be inactivated in these particular cells. Okay, so this is the first case where the X inactivation occurs in the before the pre-implantation female mammalian embryo. Okay, so this is correct option. Then one of the two X chromosomes in each diploid cell of a normal woman is randomly selected to undergo a X inactivation and becomes highly condensed. So yes, so as we discussed just now, that one either of the X chromosome, this is paternal and this is maternal. So either of these chromosome is inactivated and this becomes highly condensed because of the excess RNA. Okay. The process involves epigenetic silencing of each gene on one of the two X chromosome, either the maternal X chromosome or the paternal X chromosome. So yes, so we need the methylation of the CpG island or the LNC RNA. Both of these will be considered as an epigenetic screen. So that's why uh, the process of X inactivation requires the epigenetic screening of each gene on one of the two X chromosomes. Okay, so this option is also correct. Once the decision is made to inactivate a paternal or a maternal X chromosome in a cell, all descendant cell will continue with that pattern of X inactivation. No. Why? Because this is cell specific. So it cannot be carried forward. It will not be carried forward from one cell to its daughter cell. Okay, so it will be with that particular cell only. In a daughter cell, the either the paternal or maternal X chromosome will be inactivated. Okay, so this is not the case. So the incorrect statement out of these is fourth one. Okay, so this would be the correct answer for the question. Okay, now moving on to the next question. X chromosome inactivation in mammals is mediated by. So this is as in DBT bet paper in part A. So the X chromosome inactivation in mammals is mediated by microRNA, long non-coding RNA, messenger RNA, or short RNA directed methylation of histones and DNA. So the correct answer is long non-coding RNA, and the name of the RNA is exist RNA. Okay. So you guys must also remember the size of this particular RNA. So just check for that. That might also be asked as a direct question. So answer is 2. Now moving on to the next question. Following are few statements with respect to the isoacceptant tRNA and isodecoded tRNA. Which of the following is correct? So, before starting this question, I have this we have not covered in our lectures. So, what is iso-accepting tRNA? So, iso-accepting tRNA are basically the tRNAs that have different anticodons for the particular codon but carry the same amino acid. So, it will be, for example, we have um, here we have UUU, and here we have A A. So this is the anticodon, this is the tRNA, and this is the mRNA. Okay. So this particular codon will code for, so it will have a tRNA that codes for phenylalanine. Okay. Now this particular uh, based on wobble hypothesis. Okay. So the third base uh, in the codon can be um, like freed for the complementary base pairing between the different bases. So due to wobble hypothesis, this will be free, and the same tRNA that we have for the previous codon, this will be able to bind this also. Okay, so this is the second anticodon that is different, but the tRNA that is using it is the same one. Okay, so this is the iso accepting tRNA. Okay, so here the tRNA is same for both for this particular codon and this particular codon. Okay, so that's the case, and the amino acid is same. And then we have the iso decoder tRNAs. So what happens in that case? The tRNA is same for the same anticodons. Okay, so the tRNA is like for UUU, it will be for the anticodon will be same, similarly with the other, but the body of the tRNA will be different. So other than the anticodon loop, the other base pairs will be different with as compared to the other tRNA. Okay, so this is the two types of tRNA that have been like studied in the cell. One is the iso accepting tRNA and the iso decoder tRNA. Okay, so we'll search for the correct options based on the description that I have given. So iso accepting tRNA charged with the same amino acids but have different anticodons. Okay, this is correct. Whereas isodecoder tRNAs have same anticodons but different body sequences. So yes, this is correct. Then we have the second option. Isoaccepting tRNAs charged with different amino acids but have the same anticodons, whereas isodecoder tRNAs have the same anticodons but different body sequences. So this is wrong. Okay, so because this has the same, so sorry, Uh, so this is the second option that is wrong. Okay, because this is different amino acids but with the same anticodons. Okay. Then we have the isoaccepting tRNAs charged with the same amino acid but have different anticodons but different body sequences. So this is only about the isodecoder tRNAs as compared to the isoaccepting tRNA. Then we have isoaccepting tRNAs charged with the same amino acid but have same anticodon. So this is the case with the isodecoder tRNA, and whereas isodecoder tRNAs have same anticodons but different body sequences. So this option is also wrong. So the correct option is only the first one. Okay. Now moving on to the next question. This thing we have missed earlier when I was explaining the protein synthesis. So how did the scientists reach to the three codon? So that was what I explained was the calculation. Okay, so we had four bases. So based on that, three would be sufficient. Would give 64. That would be sufficient for the number of amino acids that we have for the codon for coding. So how did the scientists reach for this particular number based on the experiments? So scientists like Nierenberg, Hargobind Khurana, they performed various in vitro translation assays. So what is in vitro translation? Now, as you know, for translation, what do we require as a raw material? So we requires mRNA, that based on which the amino acid will be coded. Then we require ribosome. Okay, we require tRNA with different amino acids. Okay, then we require the tRNA synthetase, amino acid synthetase. Okay, then we require different IFs. IF, uh, for prokaryotes we require three IFs. For eukaryotes we have multiple IFs. Then we require EFs and release factors. Okay, and lastly we require GTP, Mg2 plus, and all those stuff. Okay, so these are the things that are required during the uh, translation cycle. So they performed an in vitro translation assay in which so in which they used a bacterial extract containing all these components, because con- purifying all these components would be difficult. So they prepared a cell culture extract. So bacterial cell culture extract is there. Okay, so they prepared this extract, and we have the mRNA alone. So, to this extract, they added the different mRNAs that code for different amino acid. For example, we have a stretch of U that will only code for polyphenylalanine. Then we have poly A that code for polylysine. We have C over in code for polyproline. Okay, so we have all these three kind of structures, and what they found was that yes, instead of the initiating amino acid being methionine, the like what they got was different proteins. For example, this mRNA coded only for polyphenylalanine. This mRNA with poly A code for polylysine, and here it coded for polyproline. Okay, so that's why they devise certain other mechanism. For example, they mutated certain bases, like, and used this kind of sequence, so that they would know that which amino acid would be next be encoded in the chain. And similarly, for other they used triplet codons also. Okay, so that would give us an idea that whether three letter is being used for making the like translating an mRNA to the protein, or two-letter code is used. Okay, so this is how the scientists designed, and they used it for determining the codon part. So the triplet codon is being made with the three bases. Okay, so based on these in vitro experiments, they identified that okay, so the codon is a three base sequence that codes for one amino acid. Okay, so why I discuss this now? Because of the next question. So the next question states a synthetic mRNA of repeating sequence 5 prime CACACA, okay, till the end, is used for a cell-free protein synthesizing system like the one used by Nirenberg. Okay, just now we explained how it was done. If we assume that protein synthesis can begin without the need of an initiator codon, what products would you expect to occur after protein synthesis? Okay, so we have an mRNA sequence that is a repeat of this CA in uh, multiple times. Okay, so if there would be codon, codon is a triplet, so that codes for CAC, then ACA, then CAC, then ACA, and so on and so forth. Okay, so based on the table of codon, that how many what codon codes for which amino acid, So CAC codes for histidine and ACA codes for threonine. Okay, so based on these particular amino acid chart, the protein composition would be histidine, then threonine, then histidine, then threonine. Okay, so like this till the C terminal. So N terminal will be the starting of the protein, like the 5' terminal of the mRNA. Okay, so what are the options? One protein containing a single amino acid? No, no single amino acid will be formed in this. Okay, then we have two proteins, each with the alternating sequence of two different amino acids. No, only one protein will be formed because the initiating codon is one only. And then we have one protein with an alternating sequence of three different amino acids. No, because only the one base like alternative codon will be different. So there will be two alternating amino acids rather than three. Okay. Then we have, so the first option is wrong, second is wrong, and third is wrong. Then we have one protein with an alternating sequence of two different amino acids. So yes, here we have alternating sequence of two different amino acids, histidine and threonine. So the correct option is option D. Okay. Now moving on to the next question. Each amino acid tRNA synthetase is precisely able to match an amino acid with the tRNA containing the correct corresponding anticodon. Most microorganisms uh, have 20 different tRNA synthetases. However, some bacteria lack the synthetase for charging the tRNA for glutamine with its cognate amino acid. Okay. So what they are saying is, so most have different 20 different type of tRNA synthetases, but some of the amino acid do not have the tRNA for tRNA synthetase for glutamine. Okay, with the two code for the glutamine amino acid. How do these bacteria manage to incorporate glutamine in their proteins? Choose the correct answer. So the first option states that glutamine is not present in the newly synthesized bacterial protein. Post-translational modification converts glutamate to glutamine in all the required sites. Then in these bacteria, the amino acid tRNA synthetase specific for tRNA glutamate also charges tRNA glutamine with glutamine. Okay. Then we have in these bacteria the amino acid tRNA synthetase specific for tRNA glutamate also charge tRNA glutamine with glutamate. A second enzyme then converts the glutamine of the charged tRNA glutamine to glutamine. In these bacteria, the amino acid tRNA synthetase charges tRNA glutamate with either glutamate or glutamine according to their requirement during protein synthesis. So this was the information plus application based question. So the first option states that glutamine is not present in the newly synthesized bacterial protein. Post translation modification converts glutamate to glutamine in at all required sites. So this statement is wrong. Okay, because the amino acid that is being formed by post translation modification is selenocysteine and pyrrolysine only. Okay, so none other amino acids are formed by this PTM modification. So the first option is wrong. Then in these bacteria, the amino acid tRNA synthetase specific for tRNA glutamate are also charges tRNA glutamine with glutamine. No, this is also wrong. Why? Because the tRNA synthetase are so specific. These are specific for particular amino acid, so they cannot be used for different amino acids. Okay, so tRNA synthetase glutamate cannot load glutamine. Okay, so this is the case with this. So this option is wrong. Then in these bacteria, the amino acid tRNA synthetase specific for tRNA glutamate also charge tRNA glutamine with glutamate. a second enzyme that converts the glutamate of the charged tRNA glutamine to glutamine. Okay, so yes, this option is correct. Why? Because the tRNA synthetase for glutamate can recognize tRNA synthetase for glutamine. This is for glutamate and this is for glutamine. So this tRNA synthetase recognizes both of this tRNAs, and what do they do? Then, after charging this glutamine tRNA with glutamate, so they charge first with glutamate, then the side chain of this glutamate is changed to tRNA glutamine. Okay, so this thing happens when the tRNA glutamine is not there. So this is the post-translation modification when tRNA glutamate is added first and then glutamine is formed on that particular tRNA itself. Okay, and in these bacteria, the fourth option, in these bacteria, the amino acid tRNA synthetase charges tRNA glutamate with either glutamate or glutamine according to their requirement during protein synthesis. Okay, so this is also wrong because of the specificity of the amino acid tRNA synthesis. Okay, so the correct option is answer number 3. Okay, now moving on to the next question. Many organisms, so it's a similar kind of question as the previous one. Many organisms encode only 18 amino acid tRNA synthetase, that is abbreviated to AARS. These organisms like AARS that use asparagine or glutamine as one of the substrates for direct amino acylation of the tRNA asparagine and tRNA glutamine respectively. Which one of the following statement represents the correct option? So the first option is the organism lacking asparagine RS and glutamine RS lack asparagine and glutamine in their proteins. In these organisms, selected aspartate and glutamate residues in the proteins are post-translationally modified by the regulated mechanism. In these organisms, the tRNA asparagine and tRNA glutamine are first amino acylated by asparagine RS and glutamate RS respectively, and then the aspartate and glutamate attached to the tRNAs are modified to asparagine and glutamine respectively. And in these organisms, the precursor of mRNA can be encode aspartate RS and glutamate RS are respectively or alternatively spliced to generate asparagine RS and glutamine RS. So, like the previous question, some of the bacteria do not have glutamine RS and asparagine RS. Okay, based on their complexity. So that's why what happens, the similar concept will apply here. So the glutamate RS will load glutamine uh, tRNA with glutamate, then post translationally this will be converted to glutamine. Okay. Similarly, aspartate RS will load asparagine tRNA with aspartate, and then post translationally this will be converted to asparagine. Okay, so this will happen in both the cases, like the previous question. So the correct option would be the organism lacking asparagine RS and glutamine RS lack asparagine and glutamine in their proteins. So this is wrong. Then we have in these organisms, selected aspartate and glutamate residues in the protein are post translationally modified by a regulate mechanism. No, this is also wrong because how can you distinguish between aspartate and asparagine then? Okay. In these organisms, tRNA asparagine and tRNA glutamine are first amino acylated by aspartate RS and glutamate RS respectively, and the aspartate and glutamate attached to the tRNAs are modified to asparagine and glutamine respectively. So yes, this is the correct answer. So the correct answer is option number 3. This was similar to the previous question. Okay. Now moving on to the next question. Ribosome prepared from a bacterium were fractionated by sucrose density gradient centrifugation. So what does sucrose density gradient centrifugation does? Whenever, for example, you have different protein components and you have a medium that is sucrose based. So when you run this particular at a particular RPM, so what it does is it will create a density gradient around the cell. Okay, so that would create, well, like the proteins will be distributed among the protein will be distributed among the various densities based upon their molecular weight and their densities. Okay, so the lowest one will have the highest density, followed by the uh, following the descending order. Okay, so this is the case. Now, how this is how the ribosomes are separated. Okay, to separate the 30s, 50s, and 70s population of the ribosome. Okay, so when ribosome preparation was incubated individually with either elongation factor EFG or a newly identified protein X or GTP, the profile remained unchanged. So they used different permutation combination with the EFG and a protein X and GTP, and they found that the profile remained unchanged. Okay, so in the first option, okay, then we'll carry on. Likewise, no changes were seen in the profile when the ribosomal preparation was incubated with EFG plus GTP or protein X with GTP. Okay. However, when the ribosomal preparation was incubated with the protein X, EFG, and GTP together, it resulted in a change of profile which showed a decrease of the 70s peak area and increase in the peak area of 30s and 50s, that is shown in panel 2. So panel 1 suggests when they have incubated with EFG plus GTP or protein X plus GTP. So they are different in both the cases. So here the pattern remains: the 70s peak is highest, then the 50s, then the 30s. Okay. So here the 70s association is more as compared to the subunits of the ribosome. Okay. Now, when this particular fraction is incubated with all the three proteins, EFG, GTP plus protein X, so in that case, what happened? The 70s wala peak was a bit reduced as compared to the 50s and 30s wala peak. Okay, this was panel 2. So choose the option that defines the correct conclusion from the observation. So protein X is an anti-association factor which functions in the presence of EFG and GDP. Protein is X. Protein X is a dissociation factor which functions in the presence of EFG and GTP. Protein X binds GTP. Okay. So in the question, based upon the first option. So here the 70s is associated in the presence of X, GTP, EFG, or GTP. Okay, so this protein will not be an anti-association factor. Okay, because it cannot inhibit the association of the 70s. Okay, so the first option is wrong. The second option, the protein X is a dissociation factor which functions in the presence of EMG and GTP. So yes, this option might be correct because here, when all the three were there, the EFG, GTP, and X, then in that case, 70s peak is reduced as compared to the 50s and 30s peak. Okay, so here it would act as a dissociation factor. Okay. Now the third option, protein X binds GTP. No, this is not it has been not mentioned in the question because here when the protein X was with GTP, then it cannot function alone. So it cannot be concluded from this data that protein X binds GTP or not. Okay. And EFG is known to bind GTP, hence it can be concluded that the effect of GTP is through EFG and protein X does not bind GTP. So the same reason here we cannot conclude whether X protein binds GTP or not. Okay, might be the EFG can bind to GTP and X cannot, So this option is also wrong. Okay, so the correct option is option 2. That is, it acts as a dissociation factor in presence of the EFG and GBT. So option 2 is correct. Okay, so the correct answer is option 2. Now moving on to the next question. In mammals, CGRH sequences are usually methylated at C. Okay, so they are talking about the CPG islands. Okay, so in this, the C is methylated. Although the promoters of housekeeping genes are often associated with CPG islands, yet they are expressed in mammals. Which one of the following best explains it? So, here they are saying that housekeeping genes also has CPG island in their promoters, but then also these were expressed. Okay, so what are the options? Methylation of cytosine does not prevent the binding of RNA polymerase 2 with the promoter, so housekeeping genes are expressed. During housekeeping gene expression, the enzyme methyltransferase is temporarily silenced by microRNA, thus shutting down the global methylation. Third option, unlike within the coding region of a gene, CG-rich sequences present in the promoters of active genes are usually not methylated. And fourth option, as soon as the cytosine is methylated in the promoter region, the enzymes of DNA repair pathways remove the methyl group, thereby ensuring gene expression. Okay, so now we'll see the explanation of different options. Methylation of cytosine does not prevent the binding of RNA polymerase 2 with the promoter. So, housekeeping genes are expressed. So, this option is totally wrong conceptually. How? If the CPG island is methylated, the transcription factor will not be able to bind the promoter. Hence, RNA polymerase 2 cannot be recruited. So, there will be no expression of the gene. Okay, so here methylation of cytosine does not prevent the binding of RNA pol 2 with the promoter, and so housekeeping genes are expressed. So this is the explanation. So this option is wrong. Then during housekeeping gene expression, the enzyme methyltransferase is temporarily silenced by the microRNA, thus shutting down the global methylation. So this is also not correct. Okay, because only the housekeeping gene expression is being regulated. Okay, the other gene expression is already there, or other gene expression is also being regulated like this only. Okay, so the global methylation cannot be shut down at a single stretch. Okay, because that will hamper the full process of transcription and expression of the gene, because that will lead to the expression of some genes that are not involved at that particular time of development. Okay, so here there will be the specific microRNA can inhibit specific methyl transferase, that is a different story, but the global methylation cannot be affected. So this option is also wrong. The third option, unlike within the coding region of a gene, CG-rich sequences present in the promoters of active genes are usually not methylated. So yes, whenever the promoter region where the CPG island is there, so this is an active promoter, this will be not methylated. Okay, this would lead to the expression of the gene. So this is the way that how the expression is maintained. Okay, so this can be the correct option. Then as soon as the cytosine is methylated in the promoter region, the enzymes of DNA repair pathways remove the methyl group, thereby ensuring gene expression. No, this is also wrong, because the methylation is not removed simultaneously. Okay, so it will take some signaling pathways to remove this methylation. So this option is also wrong. So the correct option is option number 3. Okay, so the correct option is option number 3. Now moving on to the next question. So the next question is, which of the following is true regarding the mRNA for translation in prokaryotes and eukaryotes? mRNA is polycystonic in prokaryotes and has extensive mRNA processing, whereas mRNA is monocystonic in eukaryotes and has little mRNA processing. So the second option, mRNA is polycystonic in both prokaryotes and eukaryotes, whereas extensive mRNA processing occurs in eukaryotes and little mRNA processing is prokaryotes. Then mRNA is polycystonic in prokaryotes and has a little mRNA processing, whereas mRNA is monocystonic in eukaryotes and has extensive mRNA processing. Then mRNA is monocystonic in both prokaryotes and eukaryotes, whereas extensive mRNA processing occurs in eukaryotes and prokaryotes both. Okay, so first discuss the uh, difference between the prokaryotes and eukaryotes. So the mRNA is polycystonic in prokaryotes. So this is the operon concept. Here comes the operon concept. And in eukaryotes, it's monocystonic. So one gene, one enzyme, one gene, many enzymes. Okay, so this is the case. And then in prokaryotes, the mRNA processing is less extensive. And here the mRNA processing is more extensive. Okay, so these were the differences. Now we'll see the options again. mRNA is polycystonic in prokaryotes and has extensive mRNA processing, whereas mRNA is monocystonic in eukaryotes and has little mRNA processing. So the type of RNA is correct. In prokaryotes, is polycystonic and in monocystonic in eukaryotes. But the mRNA processing is less extensive in prokaryotes as compared to eukaryotes. So the first option is wrong. Moving on to the next option, mRNA is polycystonic in both prokaryotes and eukaryotes. That is conceptually wrong because in prokaryotes it's polycystonic, but in eukaryotes it's monocystonic, and extensive mRNA processing occurs in eukaryotes and little mRNA processing in prokaryotes. That is correct, but the option is wrong. mRNA is polycystonic in prokaryotes and has little mRNA processing, okay, whereas mRNA is monocystonic in eukaryotes and has extensive mRNA processing. Yes, this option is correct. And the last one, mRNA is monocystonic in both prokaryotes and eukaryotes. No, in prokaryotes it's polycystonic and in eukaryotes it's monocystonic. Whereas extensive mRNA processing occurs in both eukaryotes and prokaryotes. So this is also not correct. So the correct option is option number C or option number 3. Now moving on to the next question. So all of the following are true regarding translation initiation in prokaryotes and eukaryotes except the ribosome small subunit binds to Shine-Dalgarno sequence in prokaryotes, whereas ribosome small subunit binds to the 5' 7-methyl cap, 7-methyl guanosine cap of eukaryotic mRNA. Then ribosome small subunit binds to Shine-Dalgarno sequence in prokaryotes, whereas ribosome small subunit binds to the Kozak sequence of the eukaryotic mRNA. Then Shine-Dalgarno sequence is a conserved sequence in prokaryotic mRNA, whereas Kozak sequence is a conserved sequence in eukaryotic mRNA. In both prokaryotes and eukaryotes, the ribosome large subunit binds only after binding of the small subunit with uh, mRNA. Okay, so let's go back and revise what was the initiation in prokaryotes. So first, prokaryotes. So what happens? This is the mRNA. Here we have the Shine-Dalgarno sequence. This is bound by the small subunit of ribosome that have 16S RNA. Okay, so this recognizes the Shine-Dalgarno sequence in prokaryotes. This is the binding of the small subunit with the mRNA. Then in eukaryotes, we have two things. First is the 5 prime methyl cap, methyl guanosine, and then we have the Kozak sequence. Okay, so now the small subunit that is 40S, 18S ribosome recognizes Kozak sequences, and some other protein recognizes the 5 prime methyl cap. Okay, so this is the initiation in both prokaryotes and eukaryotes. Now we look for the options again. So ribosome small subunit binds to Sharon-Dalgarno sequence in prokaryotes, whereas ribosome small subunit binds to the 5 prime 7 methyl guanosine cap of eukaryotic mRNA. No, the prokaryotes wala part is correct, but the eukaryotic mRNA part is not correct. Okay. Then ribosome small subunit binds to Shine-Dalgarno sequence in prokaryotes, whereas ribosome small subunit binds to the Kozak sequence of the eukaryotic mRNA. So yes, this option might be correct because the small subunit of ribosome binds to Shine-Dalgarno sequence in prokaryotes, whereas the small subunit in eukaryotes bind to Kozak sequence. Then Shine-Dalgarno sequence is a conserved sequence in prokaryotic mRNA, whereas Kozak sequence is a conserved sequence in eukaryotic mRNA. So this is correct to some extent. Okay. Then in both prokaryotes and eukaryotes, ribosome large subunit binds only after binding of the small subunit with the mRNA. So this is also correct. Okay, so option C is also correct and option D is also correct. Now we have to identify the wrong statement. Okay, so option A is the wrong statement among the following statements. Okay, so the correct answer is option 1 or option B. Now moving on to the next question. In order to ensure that only fully processed mature mRNAs are allowed to be exported to the cytosol, pre-mRNAs associated with the snRNPs are retained in the nucleus. To demonstrate this, an experiment was performed where a gene coding a pre-mRNA with a single intron was mutated either at the 5' or 3' splice site or both the splice site. Okay, so here during the transport, as we have seen, the fully processed. So this is the primary RNA which have subsequent introns in between the exons. This is processed, so spliced, and this is the matured RNA. This is transported to the cytosol. Okay. Now, only the mature mRNA is only transported to cytosol. For that, the pre-mRNA associated with the snRNPs are retained in the nucleus. Okay. To demonstrate this, an experiment was performed where a gene coding a pre-mRNA with a single intron. So here they constructed a gene that have only one intron, are mutated either at the 5 prime or the 3 prime splice site. So this is the 5 prime splice site and this is the 3 prime splice site. So either of them is mutated or both the splice site. Okay, so both of them can also be mutated. Given below are few possible outcomes. Pre-mRNA having mutation at both the splice sites will be retained in the nucleus because of the presence of bound snRNPs. So yes, uh, so the mutation at the 5 prime or the 3 prime splice site. So as there is any mutation in the 3 prime or 5 prime splice site, there will be no binding of the snRNPs. Okay, because this site is mutated, the proteins or the RNA cannot recognize this particular site, so they will not be bound to that particular intron. Okay, so the first option, pre-mRNA having mutation at both splice sites will be retained in the nucleus because of the presence of bound snRNPs. So this is wrong because the snRNPs would not be able to bind to this particular mRNA. Then pre-mRNA having mutation at both the splice sites will be exported to cytosol because of the absence of bound snRNPs. So yes, this is correct because the snRNPs will not be bound here. So if there is no bound snRNPs, then this would be exported to the nucleus. Okay. So in the third case, pre-mRNA mutated at either 5 prime or 3 prime splice site will be retained in the nucleus because of the presence of bound snRNP. The same explanation, because it will not be able to bind the 3 prime or 5 prime splice site, hence it will be exported. Okay. So this is also wrong. Then the pre-mRNA mutated at either 3' or 5' splice sites will be exported to the cytosol because of the absence of the bound snRNPs. So yes, the it will be transported because the snRNPs will not be able to bind here. Okay, so this is the case. So the correct combination would be B and D. So here B and D is option present in the option 3. So the correct answer would be option 3. Okay. Now moving on to the next question. Polysome profiling of cells treated with three hypothetical translation inhibitors is shown in the plots below. The three inhibitors are CHP, that is leaky inhibitor of translation, LTM, that arrests ribosome at the initiation codon, and PTM, that inhibits ribosome scanning. Okay, and these are the graphs that are being shown. So this is the 40s, 60s, 80s, and the polysome. Then 40s, 60s fraction, and then the 80s, but the polysome fraction is not there. And in this, the 40s and 60s is there, but the 80s fraction is not there. Okay, so now CHP is a leaky inhibitor of translation. So CHP is a leaky inhibitor of translation. So what will happen if CHP will be applied here? Then that will allow the ribosome to be bound to multiple sites. Okay, so then polysome will be formed here in the eukaryotic system. So the first option, that is the CHP, will be linked to A. Then we have LTM that arrests the ribosome at the initiation site. So what will happen if the ribosome is bound to this particular mRNA, then it will not be able to move forward. Hence, polyribosome will not be formed. Okay, so here only three fraction would be there: 40s, 60s, and 80s. Okay, so the second option correspond to B. Then we have PTM that inhibits ribosome scanning. Okay, so how does ribosome scan? First, the small subunit binds. Okay, then it scans the Cossack sequence and position it there, finding the first initiation codon. Then the 60th subunit comes. Okay, so this comes first and this comes second. So if PTM is applied here, then the ribosome scanning is inhibited. So here, what will happen? It will remain bind to the 40s, not the 60s. Hence, 80s will not be formed here. Okay, so in the last one, ATS will not be there. So the third option correspond to the C option. Okay, so the here the correct options is option number one. Okay, because it relates to all the three combinations. Okay, yes, the correct option is option number one. Now moving on to the next question. Which one of the following statements is not correct? Together with proteins, rRNA provides a site for polypeptide synthesis. Okay, so the RNPs, ribosomal RNPs, are basically involved in protein synthesis. That is true. Uh, all DNA molecules are unbranched polymers of nucleotides. That is also true. Okay, the DNA is made up is polynucleotide, and this is unbranched. DNA is synthesized in 5 prime to 3 prime direction, while RNA occurs in 3 prime to 5 prime direction. So no, both DNA and RNA synthesis occurs in 5 prime to 3 prime direction. Okay, a tRNA anticodon may pair with more than one codon. Yes, this is what this is called. You have to comment me in the comment section. Okay, so a tRNA anticodon may pair with more than one codon. So what this is called? Okay, so the correct option is option number three. Sorry, it is not the correct option, but as per the question, we have to identify which is not correct. So option three is not correct. Okay, so the correct answer is option number three. Now moving on to the next question. Presence of selenocysteine in proteins in E. coli is a consequence of. Okay, so selenocysteine is present in generally prokaryotes, that is made by the post-translation modification of the cysteine molecules. Okay, where the sulfur is replaced by the selenium metal. So in cysteine, the sulfur is replaced by selenium. Okay, so the first option is post-translation modification of cysteine present in special structural regions of the protein by cell B and cell C. Port PTM of serine present in special structural region of the proteins by cell B and cell C. Aminoacylation of special tRNA synthetase, tRNA selenocysteine by serine tRNA synthetase with serine, followed by further modification of the attached serine to selenocysteine, followed by the transport to the ribosome by cell B, and aminoacylation of a special tRNA by serine tRNA synthetase with selenocysteine, followed by its transport to ribosome by cell B. So, um, as we know, when we see the structure of serine, so how does it look? This is the alpha anomeric carbon. We have H, we have NH2, we have COOH, and here we have CH2OH. So this is the serine molecule. Then we have cysteine molecule. Here instead of O we have S. Okay, and in selenocysteine we have the selenium molecule instead of the S. Okay, so this is the basic difference between serine, cysteine, and selenocysteine. So sometimes what happens, the prokaryotic system forms selenocysteine from serine, not from cysteine. Okay, so the first option states that PTMs of cysteine present in special structural region of the proteins by cell B and cell C. So no, this doesn't happen inside the cell. Then PTM modification of serine present in special structural regions of the proteins by cell B and cell C. No. Okay. Then we have amino acylation of a special tRNA selenocysteine by serine tRNA synthetase with serine followed by further modification to the attached serine to selenocysteine followed by its transport to the ribosome by cell B. So yes, this happens in the correct way. So what happens? The tRNA, serine tRNA synthetase basically load tRNA, selenocysteine tRNA with serine, and here it is modified to selenocysteine, and then it is transported to cytosol by cell B. Okay, and then amino acylation of a special tRNA selenocysteine by serine tRNA synthetase with selenocysteine followed by its transport to ribosome by the cell B. No, so this is the correct answer. So option 3 is the correct answer. Okay. Now, moving on to the next question. In an experiment, it was observed that a protein was upregulated in the cancer tissues compared to control tissues that showed correlation with disease progression. Following are few possibilities which can explain the above observation. So, we have control tissue and we have cancer tissue. So, this protein is normal in this particular control case, but it's higher in the cancer tissues. Okay, so this is the part of the disease progression. Now, the observations are a mutation could be located in the 3' UTR of the corresponding mRNA at a miRNA binding site. A mutation changes the conformation of a protein, resulting in better stability. A mutation in the corresponding mRNA promotes ribosome breakthrough of the termination codon, resulting in increased synthesis of the protein. And a mutation in the corresponding mRNA increase the stability of the RNA due to change in the secondary structure. Okay. So this is the mRNA. Here we have the 3' UTRs, that is untranslated region, that regulates the process of translation. This is the major structural gene, and we have the 5' UTR region before the termination cell. Okay, so the observations are the mutation in the 3' region. Here the it is mutated, so it leads to the mutation in the microRNA binding site. So what this means is that the microRNA cannot bind to this particular site, and hence it will not be able to regulate the protein synthesis. So this protein is forming in large quantities in the cancer tissue, not in the control. Okay. So yes, this can be a possibility. So first option is correct. Then, any mutation that changes the conformation of the protein for better stability will lead to better activity. Okay, not the levels. So, when if either of the mutation is present here, that leads to better stability, but not the number of the protein molecules. So, this option is not correct. Option B is not correct. Then a mutation in the corresponding mRNA promotes ribosome read through of the termination codon, resulting in increased synthesis of the protein. So yes, this can happen because when the termination codon is mutated, so there is a mutation in the mRNA that promotes the read through of the termination codon, so that 5' UTR is also dysregulated. Okay, so this would lead to the increase in amount of the protein because here the regulation cannot happen because of the uh, mutated 5' UTR. Okay, so this option can be correct. A mutation in the corresponding mRNA increases the stability of the RNA due to change in its secondary structure. So this might be the possibility because if the RNA is stable, then it will be like not prone to degradation by the uh, nucleases. Okay, so the secondary structure is stable, then this would not happen. Okay, so a mutation leading to stable secondary structure of the RNA can be helpful to prevent the degradation by the nucleus. So this option is also correct. So the correct option is A, C, and D. Okay. So based upon the options, we have C, D, and A in the third option. So this is correct. So the correct answer is option C. The next question, in trypanosome, a 35 base leader sequence is joined with several different transcripts, making functional mRNAs. The leader sequence is joined with the other RNAs by a specific RNA ligase, the process of transsplicing, a nucleophilic attack caused by a pre-guanine nucleotide, and a nucleophilic attack caused by 2' OH of the internal A present in the leader sequence. So we have discussed this before also. In trypanosomes, we have a process called as trans-splicing. Okay, so here this is the first part of the mRNA. This is 5' and this is called as a leader sequence. It has a 5' splice set, and the other part, that is the 3' here, is the 3' splice set. Okay, so now here the leader sequence will help to the do the splicing, and then the mature mRNA will be formed by the process of trans splicing. Okay, so this happens in trypanosoma only. So here the correct answer is option B. So second option. So now moving on to the next question. In prokaryotes, the initiator tRNA is first charged with methionine, followed by the addition of a formyl group to the methionine by the enzyme methionine tRNA transformylase, Given below are several statements in this context. Uh, all prokaryotic proteins have formyl methionine in their amino terminal end. Okay, so in prokaryotes, the first amino acid is formyl methionine. Okay, followed by different amino acid, and this is the towards the N terminal side. Okay, so the first option, all prokaryotic proteins have formyl methionine at their amino terminal end. So this is correct. The first amino acid is formyl methionine. Deformylase remove sorry, deformylase removes the formyl group from amino terminal methionine. No, this does not like happen because the formyl methionine is the recognition for the first amino acid. Okay, so here the deformylase never removes the formyl group from the methionine. All prokaryotic proteins have methionine at their amino terminal. All prokaryotic proteins have methionine at their amino terminal end. So this is also not correct because all prokaryotic proteins have formyl methionine at their end. Aminopeptidases often remove the amino terminal methionine. No, aminopeptidases remove peptides, amino acid from the N terminal. So it is not specific for methionine. Okay, so this can also be false. Aminopeptidases remove amino terminal formyl methionine. So this can be true. Okay, so the uh, first one is the formyl methionine. So the aminopeptidases will remove the formyl methionine as compared to normal methionine, and all prokaryotes have formyl methionine at their amino terminal end. So the correct option is option number two. So sorry guys, here is a correction. So what happens during translation? Although the first amino acid that is initiated is formilated methionine, but after the translation happens, this formilated methionine is either the methionine is removed or the formyl group is removed. Okay, so it does not remain in the prokaryotic proteins. So this is the case with the prokaryotic proteins. So the first option, now we'll see the options again and then we'll deduce the answer. So all the prokaryotic proteins have formyl methionine at their amino terminal end. So this option is wrong because the formyl group or the formilated methionine is only removed from the proteins. Then deformylase removes the formyl group from the amino terminal methionine. So yes, this is correct. All prokaryotic proteins have methionine at the amino terminal end. So based upon this fact, if the formilated methionine is removed, then this is option is also wrong. Then aminopeptidase often remove the amino terminal methionine. So when we talk about the aminopeptidase, so if the amino acid like the amino terminal methionine is there, then it would be removed. So yes, this option can be correct. And then E aminopeptidase remove the amino terminal formyl methionine. So this option can be wrong. Okay, so the correct option is option B and option D. So that is fourth option. So here the correct option would be option 4. Okay, now this was the last question for today, and here we come with the end of this RNA biology lecture series. So, today it was a delay in the class, So if you guys have any doubt, you can contact me via mail. So you guys have my mail ID, and I'll like respond you whenever I'll be free, and we'll take it forward from there. So thank you guys for your patience and for joining till this end of the RNA biology lecture series. So thank you.